Hello everybody, my name is Michael Stab and I'm going to present to you Subproject 6, which I will head together with Heike Feldhaar, who you know already from Subproject 1. Our Chinese partner project will be headed by Zhaojun Liu from the Institute of Botany of the Chinese Academy of Sciences. Past research has shown that there is often strong crosstalk and congruence between plants and consumers, meaning that when the number of plant species in an ecosystem increases, as shown here on the x-axis, the number of arthropod species also increases. This has been shown initially by grassland research, but also for forests as a synthesis project, comparing data from the BEF China and the Jena experiment also have shown. Subproject 6 will focus on ants. You may now rightly ask why such a focus on ants. Ants are not only very interesting organisms, but they're also very abundant in subtropical forests, meaning they have lots of interactions and contribute to lots of functions. For example, if in such a forest you would put all ants on one side of a scale or on mammals on the other, the picture would almost always look like this. Ants have also, as a taxon, as a group, a very broad trophic niche, meaning they occur across trophic levels from being almost herbivorous to being highly predaceous, as this figure from a study conducted in Queensland in Australia shows. Let's focus on the delta 15N axis, the y-axis. Delta 15N is the heavy form of nitrogen, which can tell you a lot about the trophic position of a species in a food web. So if a species has a lower delta 15N signature, it's more herbivorous. If it has a high delta 15N signature, it's more predaceous. And species in this study are indicated by the triangles here. And if you just compare it to the pictograms of the other organisms, you see that there are ants that range from being herbivorous to being highly predaceous. Within an ant community, this is the same ant community as shown in the previous slide. There can be species that have a very broad trophic niche. Here, the delta 15N ratio shows that they occur in at least 2 trophic levels, while there are also species with a very narrow trophic niche in terms of nitrogen, as this species. So, to sum up, due to the trophic flexibility and to the variability in trophic behavior in the taxon ants, that can be very suitable for studying biodiversity and ecosystem functions. I have lots of experience on ants, both in China and in the BEF China experiment, and I would like to highlight one of our previous study conducted by my students Karl Skarbek and Merle Noack, where we tested whether the experimental treatment of the experiment, which manipulates plot diversity, or the heterospecific or neighborhood within a sampling point within a single tree individual determines the diversity of the ant community. What we found is that it's the plot diversity that has an influence on ant diversity measured as ant species richness and not the neighbor diversity. This study, which clearly showed that the plot scale is very important for the ant communities, was the starting point for the current subproject 6, because we don't know whether this relationship between tree and ant diversity means anything for interactions and for functions. There are lots of further knowledge gap. For example, there's surprisingly little work on ants in BEF experiments. It's unknown how tree diversity influences trait distribution within a taxon. We don't know if the influence of tree diversity on realized trophic niches and potential diversity-dependent shifts therein can be measured, and it has only rarely been tried to link trophic interactions to ecosystem functions in forest ecosystems. So Subproject 6 wants to tackle these questions in 3 work packages. The first work package will quantify the diversity and functional trait distribution of ants in all 300 core plots. The second work package will identify the trophic positions of the species and assess their trophic interactions. And the third work packages, this time conducted on the 64 VIP plots, will link work packages 1 and work packages 2 into a larger picture by measuring the ecosystem function scavenging. To conceptualize, BEF China is a diversity experiment. So we have a gradient in tree diversity coming from monocultures to mixtures of more species. Work package 1 within the experiment will quantify the diversity of ant species and measure their trait distribution. In work package 2, then we will assess trophic interactions by using standardized resource choice experiments to get a measure for trophic complementarity and redundancy, which we will then validate by measuring delta 15N. And finally, in work package 3, we will combine all those measures with an ecosystem function to test whether tree diversity and ant diversity and trophic complementarity influence ecosystem function rates. Work package 1 will use leaf litter ants collected with Winkler extraction and measure traits to test the hypothesis that ant species diversity increases with tree diversity, that trait distribution is broader when tree diversity is high, and that functional morphospace shifts towards more predaceous when tree diversity increases. In work package 2, we hypothesize that tree diversity increases the generality of resource use, both via ant diversity and via trait distribution, that there will be relatively more predaceous ant communities in more diverse plots, and that the community mean of delta 15N increases with diversity. We will do this with a standardized resource choice experiment, for which we already did some preliminary experiments. These experiments will use surrogates for carbohydrates, nitrogen, phosphorus, lipids, salt, and water. We will construct the ant resource network, calculate, for example, network redundancy, and combine this with measuring stable isotopes. The third work package hypothesizes that the ecosystem function scavenging increases with tree diversity and that this function will be mediated by ant diversity, trait distribution, trophic complementarity, and trophic position. We will do this by using two types of carrion, for invertebrates carrion and for vertebrate carrion. For invertebrate carrion, we will use fish food pellets, which have been used in standardized ant feeding assays before, and for vertebrate carrion, we will use chicken wings and measure the retrieval rate of ants over time. SP6 is very well integrated within Multitroph. We have strong links to all sub-projects, of which two are especially important for our project. One is the link to SP2 and SP3 via the stoichiometry baseline, and especially to SP2, which will give us data on the soil delta 15N baseline, and to the SPs 1 and 3, which sample with complementary collection methods, and it's very likely that they will also sample a lot of ants, and we will exchange specimens with them to increase the scale of the sample. SP6 will join up with the Chinese counterpart project led by my collaborator and long-lasting scientific friend Zhaojun Liu, and this project will be fully complementary to SP6, and we will test the influence of trees with extrafloral nectaries, which are part of the BEF China design on ants, herbivores, leaf damage, and leaf traits, because it's possible that trees with nectaries attract ants, which may facilitate tree growth when tree diversity is high. We have some initial funding secured for this project, and now I'm happy to lead over to the last presentation of today of set project 2, which will be given by Andreas Schuld and Helge Brühlheider. Thank you for your attention.